welcome to Lecto. Today we are going to discuss types of hemoglobin and the synthesis of the globin chain. So before we start, you should know the basic component of the hemoglobin. There are two basic components, heme group and the globin chain. So here you can see hemoglobin consists of two components. First one is the heme group and second one is the globin chain. There are 4 heme group and the 4 globin chains are present in the hemoglobin. Globin chain has different types and these are actually the protein. And as we know that the first of all DNA form the RNA and finally RNA convert into the protein. So this is the basic rule. DNA convert into RNA, RNA finally convert into the protein. It's mean that the globin chain is basically produced or the message comes from the DNA in the formation of the globin chain. And globin chain has different types. So we start from the DNA. So normally human body has 46 chromosome and 23 in pair. So here you can see that this is the chromosome 16. These are actually the homologous chromosome. And this is the chromosome 11. This is the chromosome 11. They are also in homologous state. Chromosome has 2 arms. One may be smaller or other may be larger. So here you can see chromosome 16 has 2 arms, P and Q. P arm and Q arm. P is basically the small arm. P is small arm of the chromosome and Q is the large arm of the chromosome. Same here that chromosome 11 has 2 arms, P and Q. P is smaller and Q is larger. So chromosome 16 has different genes on the small arm. So here you can see, and what type of genes? You can remember it by the pizza. Last words has 2 Z and A. So here you can see Z stand for zeta. Z stand for zeta genes which involve in the formation of the zeta globin chain of the hemoglobin. And next is the A. The letter A stand for alpha. Alpha. It has 2 forms, alpha 1 and alpha 2. These are basically the 2 forms of the alpha 1 and alpha 2. Before we discuss, you should again know the basic difference between the genes and the allele. Alleles are basically the component of the gene. Here you can see this zeta gene. Zeta gene has 2 alleles on the homologous chromosome. Here you can see this is the allele 1, this is the allele 2 and it forms the 1 gene. Same here you see that the alpha 1 and alpha 2. This is the 2 alleles of the alpha. These are basically the 2 alleles of the alpha. And again these are the alleles of the alpha 2. So there are 4 alleles present in the chromosome 16 of the small arms. And remember that these alpha genes help in the production of the 2 globin chain. Actually two genes are present and overall 4 alleles are present and they are involved in the formation of the 2 alpha globin chain. Next we see on the chromosome 11. Here you can see what types of gene actually present on the larger arm of the chromosome 11. So you will remember it by the enterprise geodata base. So you will remember this word, enterprise geodata base. E stand for epsilon. This is basically the epsilon. Next is the G. G stand for gamma. You will see this is actually here. This is the gamma. So in that situation there are actually two forms of the gamma. G gamma, there are G gamma and A gamma. There are two forms of the gamma, G gamma and A gamma. G glutamic acid and A alanine. So these are basically the amino acid, and their sequence, if you see that this is the gamma globin chain, and the difference is that in some gamma chain at the position 136 amino acid, these are actually the amino acid chain of the amino acid which form the gamma globin. At the position 136 there are two variants of the gamma globin chain. Some gamma globin chain carry glutamic acid at the position 136 and some carry alanine. So this is the two variants of the gamma globin chain. And next, D stand for delta. D stand for delta. And last one is B stand for beta. So it form a cluster, and this cluster is basically called this cluster. This cluster is called the beta globin cluster on the basis of the presence of beta genes. But here you can see this cluster alpha and zeta, it form a cluster which is called the alpha cluster gene. So these are basically the two cluster gene, alpha cluster gene and beta cluster gene. So next we will see what type of hemoglobin present in embryo, fetus and adult. Here you can see in the embryo two basic types of hemoglobin present. First one is the Gower 1, second one is the Portland, and third one is the Gower 2. These are basically the three hemoglobin, and in them, in the Gower 1 you will see 2 zeta and 2 epsilon globin chain. In the Portland you will see 2 zeta and 2 gamma globulin chain, and in the Gower you will see 2 alpha and you will see 2 epsilon globin chain. So these are basically the 4 types of hemoglobin present in the embryo. So these are basically, Gower has 4 globulin chain, Portland again 4 globin chain, and Gower 2 again 4 globin chain. So again basic concept is globin chain will be 4, will always remain the 4, but the globulin chain type will change according to the hemoglobin. So these are basically the hemoglobin which are present in embryo. And as we know that the embryo will convert into the fetus, then what type of hemoglobin is present in the fetus? This is actually the hemoglobin F. Hemoglobin F. Hemoglobin F has different globulin chain. So you will see 2 alpha and 2 gamma globin chains are present in the hemoglobin F, which is actually the fetus hemoglobin. And next, if we move forward, then the fetus convert or into the adult. So what type of hemoglobin present in the adult? 3 types of hemoglobin present in the adult. So hemoglobin A, hemoglobin A2 and hemoglobin F. But the quantity of the hemoglobin F in the adult is very small relative to the quantity of the hemoglobin F in the fetus. So this point is very much important. So hemoglobin A has different globulin chain. It has 2 alpha and 2 beta chain. This hemoglobin is very common in the adult, and this is very important all point of view. So 2 alpha and 2 beta. Next is the hemoglobin A2. It contains 2 alpha and 2 delta. 2 alpha and 2 delta globulin chain. And last is the hemoglobin F. Same here, 2 alpha and 2 gamma. So this is the fetal hemoglobin. So these are basically the hemoglobin which are present in the adult. Hemoglobin A present in large amount, then hemoglobin A2, and last hemoglobin F, which is actually present in small quantity. Same here, 4 globulin chains are present in each hemoglobin. And this is the normal hemoglobin. There are a lot of other hemoglobins which are present, but in abnormal situation like hemoglobin S, which is present in sickle cell anemia. So these are basically the normal hemoglobin which are present in normal individuals. So next we will move forward. We will discuss the graph which will help you in understanding of 2 things. First, variation in the quantity of the globulin chain, and next it will help you in understanding of at what level of development hemopoiesis will occur in our body. So first of all, you will see the x-axis and second one is the y-axis. We take x-axis time, and at the y-axis we will take the quantity or percentage of hemoglobin. So at the center you will see that is the birth. Before the birth, the time will be prenatal. Prenatal time is that time which is actually before the birth, and after birth the time will be postnatal. So this is the basically the times, and you will take it in weeks. So first of all, we will discuss the variation in the globulin chain with the passage of time. So with the passage of when the zygote start dividing, then with the passage of time, that the alpha globulin chain starts increase its quantity, and it's after approximately 6 weeks of the prenatal time, it will become constant and it will remain constant throughout the life. So this is the alpha globin chain. Next, if we talk about beta, in beta chain you will see in prenatal life beta is quantity is approximately very low. With the passage of time, its concentration increase, and after birth, their concentration increase rapidly, and this is the case of beta. This is the beta situation. And next is the gamma. In gamma situation, gamma is high in the prenatal and it will reduce afterwards. So this is the case of gamma. Gamma is high in prenatal life and in postnatal its quantity decrease. Its mean. This point is very much important. Gamma globulin chain converts into the beta. So here you can see this is the cross link. Gamma globulin chain quantity is high in the prenatal life and decrease in the postnatal life. But the beta is opposite to that of the gamma. In beta case, in prenatal life, beta quantity is low. But with the passage of time in the postnatal life, its quantity increase. So it's mean that the gamma is switched off into the beta. So this is the switch from fetal to hemoglobin. Fetal to adult hemoglobin. As we know that the fetal hemoglobin has two alpha and two gamma. But the adult, if we see adult hemoglobin, so you will see that the adult has two alpha and two beta. So you can clearly understand that the alpha will remain the same, but the conversion will be gamma to beta conversion, and this conversion is mostly occur by the BCL11A transcriptional factor. This factor is involved in the conversion of the gamma globulin chain into the beta globulin chain. And next we will see that the epsilon quantity reduce in the prenatal life, and same here the zeta will also, quantity of the zeta will also decrease in the prenatal life. And last one is the delta. So delta can occur in both prenatal, at the end of the prenatal, and it will start at the postnatal life. So if we talk about the hemopoiesis, at what level, in what organs of the body hemopoiesis actually occur? So first one, in the prenatal life, the first organ of hemopoiesis is the yolk sac. In the early stages, hemopoiesis occur in the yolk sac. And next, with the passage of time, it will convert into the liver. Next organ will be the liver, and if we move further, then it will occur in the spleen. Third organ will be the spleen. And after birth, the hemopoiesis will convert or will refer to the bone marrow. So last is the bone marrow. And throughout the life, the hemopoiesis or the formations of the blood cell will remain the function of the bone marrow throughout the life. And the hemopoiesis which occur in the bone marrow is called medullary hemopoiesis. And throughout life, you will not see the hemopoiesis in the yolk sac, liver, spleen. No, not at all. But in some situation, in disordered state or dysfunctional, in some diseases, the hemopoiesis of adult person, if there is an adult person, and you will see the hemopoiesis occur in the liver and the spleen. Yes, it can happen, but in some diseases. In disease situation, hemopoiesis can occur in the liver and spleen. But normally, hemopoiesis will occur only in bone marrow. If the hemopoiesis occur in the bone marrow, it is called the medullary hemopoiesis. But if it occur in liver and spleen of the adult person, this is abnormal situation, and this is called extramedullary hemopoiesis. So this is what organ are involved in the hemopoiesis. Next, last is the globulin synthesis. How globulin synthesis happen in the DNA. So first of all, you will see the structure of the DNA. Here you see this is the structure of the DNA. And DNA has two basic components, exon and intron. You will see that the globulin synthesis is specifically, if we take an example of beta globin synthesis, you will see that there are three exon. Exon 1, exon 2 and exon 3. These are basically the three exon or coding region. These DNA regions are involved in the coding, and there are two intron region. Here you will see this is the intron 1 and intron 2. This is 1, this is 2. There are basically the two intron. These are basically the non-coding region. Exon is coding region and intron is non-coding region. As we know, as we discussed, the DNA converted into the RNA, RNA convert into the protein. So you will see that the DNA transcription, this step is transcription, conversion of the DNA into the RNA. And in that situation, pre-RNA is produced. Pre-RNA is produced. In this situation, exon is also convert into the RNA, exon portion. Intron portion also convert into the RNA. But after that, when the pre-RNA is produced, after that a process which is called splicing happen in the pre-RNA. In that situation, the intron region will excised. These are removed, and finally the 3 exon region. You will see 3 exon region, exon region 1 of the RNA, 2 and 3. These 3 exon region will combine to form a single mature RNA. It will result into the formation of mature RNA. So after that, you will see the mature RNA. And in that situation, at the starting and ending point, cap and tail, we insert cap and tail, and it helps in the protection of the RNA from the other enzymes, because enzymes can break the RNA. So for its protection, cap and tail are added in the messenger RNA. And you will see after, when the mature RNA is produced, it will move to the ribosome. It will move to the ribosome, and at the ribosome, what process will happen? Translation will happen at the ribosome, and finally the globin will produce, which will require the beta globin chain. So this is the structure which help. This is the process. This whole process can also be called the central dogma, in which the translation and transcription two processes will occur. So, and last important point you have to remember, in intron, when there is a process continuous in the transcription, then why the RNA polymerase recognize that there is intron and there is exon? At the starting point, at the starting of the intron, you will see GT region. GT nucleotide will present in the start of the intron, and AG nucleotide is present at the end of the intron. So these are the two regions which will recognized by the RNA polymerase, and it will cut intron and excise it from the messenger RNA. So this is all about the hemoglobin types and its globin synthesis. So if you have any question, then you may ask in the comment section. Thank you.